Hello, everyone. To start with, I would really like to thank the organizers for all the effort to organize this conference in vivo and in vitro at such difficult times. However, science must go on, so I'm really glad to share with you some progress in our research. Hopefully, the title is self-explainable. So, if you look at DNA small molecule interactions, we can take two approaches. One of them is a static approach, whereby DNA is considered as a rigid template with very precisely defined structural features, and we try to find a small molecule like lock and key principle to which will fit exactly at the shape of the DNA part we are targeting. And in this aspect, one of the best examples is actually work of Peter Derwin and his co-workers over 30 years of research, which devised that if you link such heterocyclic amides into 10 to 20 meric polyamides in certain order, defined as they found out by alphabet of recognition, you see these are the heterocyclics and these are the base pairs which are recognized, then you actually can get a very high selectivity or even specificity and very high affinity of recognition of any DNA sequence you like. And it happens from the minor groove, which is actually different from the protein, which does the same recognition from the major groove. All this is based on a set of rather weak but multiple hydrogen bonds, which shows that actually, in this case, good K is consisting of a lot of weak interactions, but very precisely defined. However, what we know today is that DNAs, RNAs, and proteins are much more flexible and active in aqueous solutions under biologically relevant conditions. And binding of a small molecule does not consist of only small molecule adapting to the binding site, but goes also other way around. DNAs, RNAs, proteins change their structure to adapt to small molecules. So it can become quite different to predict what will happen because this dance actually consists of a lot of mutual interaction and probably looks like this. One of the best examples of such dynamic behavior of DNA is actually binding of the so-called threading intercalator to double-stranded DNA, whereby you can see that this part of the DNA, these base pairs, have to unzip to allow the small molecule to enter, then they have to zip again. This will not be possible if we consider DNA as a rigid, non-changeable target. So, one of the main applications of small molecules which interact with DNAs, RNAs, proteins is bioimaging. Not only one, but one of the very interesting we are interested in. And the good question is how many colors you can see. Currently, sort of record is up to 7 different colors for 7 different processes inside the cells. And even for that, you have to be lucky that none of the colors, none of dyes interacts with each other, and most of them are covalently bound. So non-covalent staining is very complicated, and this raises lots of problems, which actually push researchers to try to find new and new and new dyes. And, actually, here we come to our goal, and we asked ourselves whether this molecule dye has to be really specific or highly selective for just one target. And because of all the problems I mentioned. So actually it's maybe better if we can have one dye which will actually interact with itself in sort of aggregate when bound, and actually it will be able to bind to as many targets, DNAs, RNAs, proteins as possible with same affinity, very similar affinity, so no selectivity, but for each of them will give different spectroscopic response in different methods, of course. It's quite a challenging task. So we started to pursue this issue some years ago, as you can see, first in collaboration with Professor Carsten Schmuck. And he actually devised such molecule which resembles to the arginine, but actually it is not so similar because it has this heterocycle and some other features. Main of the feature is that actually it's curved and fits excellently into the minor groove of DNA. But when it is neutral, it does not bind. Unlikely to arginine, pK value is really acidic and you have to go in the acidic conditions to have decent binding. So it's sort of a pH driven switch on and off where you can get binding, dissociation, binding, dissociation. Affinity was not high enough, so we really did a lot of effort, as you can see here, and we tried to anchor it more strongly by adding all kinds of the chromophores. Of course, we wanted to see what is happening, and we changed a lot, really a lot of them. And the family of pyrene derivatives actually showed the most promising results. So, as usually happens, one of the first molecules we got actually was one of the most interesting. And here you can see that this one molecule can recognize simultaneously DNA and RNA by two different methods specifically. So how it works? It works in a way that when bound to the DNA, this part of the molecule goes inside the minor groove, of course under acidic conditions, and because of that gives a very strong induced CD signal in circular dichroism spectrum, this intercalator giving also small negative signal. Double-stranded RNA has completely different shape of the major and minor groove, and because of that there is absolutely zero line at the position of this signal. So CD specific signal for the DNA. But RNA has large major groove, and now this molecule forms a dimer inside, and two pyrenes form excimer, which gives very typical and very intensive fluorescent emission at about 500 nanometers. Of course, from the reasons on the previous slide, double-stranded DNA has no such signal. So fluorometric response is specific for the double-stranded RNA. So from this example and many others, which I don't have time to discuss at the moment, we actually realized that one chromophore does not mean one color. Actually, if it is possible to get him to aggregate inside the binding site of the DNA, RNA, or even a protein. And because this aggregate occupies much more space inside the binding site, it's much more sensitive to the shape. So if you have different types of double strands with different shapes of the minor or major groove, you will get different responses. Very useful property. Also, we noticed that if one has a chiral small molecule, it will form a dimer inside the binding site, which will be tilted either left or right, and because of that, it will give very, very sensitive induced CD response, either as a dimer or as a monomer if it is bound as a monomer. So you see, we don't rely only on the fluorescence. We introduced one of new methods for fine sensing, and there are also some others that pointed out that chirality also can be used as bioimaging. So far about the DNAs, but we also wanted to see what happens with proteins, and for that reason we have chosen one of the most abundant proteins in plasma. It makes 70% of protein content in plasma, and what is more important, these serum albumins are known as carriers of small molecules. They have really a lot of binding sites. So we were pretty sure that whatever small molecules we have, this is one of the best targets if we want to see if we can get the binding. Okay. So we made a lot of attempts, and we tried monomers and homodimers of two chromophores linked covalently together, but one of the most interesting recent examples is this heterodimer. This dimer is actually, these are two molecules, isosters, which differ in the linking position on the cyanide, either here or here, and this linking position controls the exposure of the positive charge to the solution. So they are, let's say, the same molecule looking quite different from the outside. Another interesting property of this molecule is that actually it is able to do fret pair, because emission of the pyrene when in interaction with aromatic shifts to 450 nanometers, and at approximately this wavelength, this cyanin absorbs, and then it can emit again at 540 nanometers. Of course, cyanin does not emit fluorescence when it is in free position, so this fret should work only when bound to the target. Okay, what happens when we add, for instance, DNA or RNA to such molecule? We get from zero extremely strong increase of the fluorescence of cyanin dye by known mechanism, how cyanin dyes work, which is expected, shows that this really is bound to the DNA. What is more surprising is actually we have almost no change in the emission of pyrene. And all these pyrene derivatives we studied before were typical for very strong emission change of pyrene upon binding to the DNA. That actually suggests that cyanin goes into the binding site of the DNA, blocks it, and the pyrene dangles outside, exposed to the aqueous solution, and therefore does not change much. However, if one excites a pyrene at its excitation wavelength, and then this pyrene emits of course emission, and then actually cyanin, which is bound inside DNA, picks up part of the energy which is emitted by pyrene, then we have efficient fret as shown here. Actually, it's not very efficient, it's less than 5%, because it's not ideal overlap of the emission of pyrene and excitation of cyanin. However, it works obviously measurably, it is measurable. When we added serum albumin to the same compounds, we got exactly the opposite response. That means that we got very strong fluorescence of the pyrene, which we associated with immersion of the pyrene inside the hydrophobic environment of the protein binding site, while intensity of the cyanin was almost negligible. We also got some fret. So then we went to the next experiment, where we mixed in one pot protein and the DNA, and we added our compounds. What we got is measuring the intensity of the fluorescence of cyanin was directly proportional to the quantity of the DNA, and also the intensity of the fluorescence of pyrene was proportional to the amount of the BSA, which meant that actually within one molecule, two chromophores agreed that one of them will go to the DNA, positively charged one, and the neutral one will go to the more hydrophobic binding site of the protein, and each of them will report the exact amount of each of the big macromolecules. This leads us to the last example in this talk, which we achieved in cooperation with Professor Todd Mather, and actually this cooperation started right here in Caparica conference 4 years ago. I was attracted by very interesting results and this quite novel shape of a molecule which had borons, which were considered until recently as non-stable in water. It has terminal 4 positive charges, good for DNA, a large hydrophobic part, good for the proteins. So we thought it might work in a way binding to both, and we agreed to start collaboration. And what happened is actually amazing. This molecule was bound very strongly in an almost nanomolar affinity to both protein and DNA and RNA. So almost no selectivity. But for all kinds of polynucleotides, it gave red-shifted fluorescence, and for the protein gave significantly blue-shifted fluorescence, which we went back to the fluorescence in the cells, and we measured the fluorescence spectrum in the cell and got a very good match to the protein fluorescence, which directly excluded DNA as the target of a small molecule inside the cell. So one molecule to bind them all. Yeah. Actually, it is possible. Of course, it has a lot of disadvantages, and it is still work in progress. But very few people work on such projects, so it is a huge playground with really not very occupied. So very interesting. So we plan to pursue not only fluorescence and circular dichroism and UV vis monitoring of this recognition, but we introduced recently also Raman and SERS sensing and some other methods where we try to actually get what we showed here. So at the end, I would really like to thank all my co-workers who did the job, and also collaborators, Professor Mader and Professor Schmuck and many others who helped us and gave us a lot of very interesting dice. Nothing goes without money, so I really would like to thank all the financial sources which supported us. And of course, nothing will have any sense if there is no audience. And for that reason, I would really like to thank you for your kind attention.